Welcome, everybody, to another Expert Talk. We're really glad you're here, and I hope that you're aware that research in ALS is continuously progressing. And at Everything ALS, we are dedicated to uncovering new discoveries, aiding in new early detection, and working towards finding a cure for ALS. You can assist us. The purpose of the speech study is to enhance the diagnosis and prognosis of ALS. We are seeking participants to provide 5 speech recordings, each lasting 15 minutes once a month for a total of 75 minutes over 5 months. We really appreciate if you could feel like that's something that you could do. We welcome healthy participants as well as individuals diagnosed or probable with ALS to join us. And if you'd like to enroll, you can go to everythingals.org/speech project. One other important project that we have is we also have another study we are actively recruiting for, the asymptomatic gene carrier study. If you are a gene carrier and asymptomatic and interested in remotely monitoring your speech, cognition, and motor skills, please contact us at everythingals.org/asymptomatic study. And with that, folks, we really appreciate your attendance tonight, and I will turn it over to Indu. Thank you, Indu.
Thank you, McFinn, and welcome everybody. And I have a great honor today to welcome Dr. Laura Ranum. Dr. Laura Ranum holds a doctorate degree and is a director of Center for Neurogenetics and Kisman Family Distinguished Professor of Molecular Genetics and Microbiology at the University of Florida. In 2011, her lab discovered that novel category of proteins are produced from repeat expansion mutation that do not require a canonical AUG initiation condone for expression. These toxic repeat associated non-AUG RAN proteins have been found in growing number of neurodegenerative diseases, including C9orf72 ALS and frontotemporal dementia. Mechanistic insights from her work have identified novel therapeutic strategies to treat RAN protein disorders. With no further ado, I would love to welcome Dr. Laura Ranum to today's event, and please take away.
Well, thank you. It's a great pleasure to be with you all tonight. It's really a big honor. I also come from a family with ALS, and I hope I don't I'm going to get choked up, but I just thought I would share that with you before I start. So, let's see.
By the way, thank you so much for sharing that. I really appreciate it. You know, all of us are here on the same mission, and thank you.
Yeah, and it's a big driver for me, and it has been. And but usually I just wear my professional hat and don't. Well, I share it offline, but not with so many people online. So
yeah. Well, we call this a citizen-driven research. So that is because we, the citizens, are all here together. All of us are affected, whether you're a researcher or not. I came in because my husband was affected, and today I'm doing my Ph.D. in ALS. So, so we all come in through different.
Yep. And my husband is affected by C9 ALS, so that's been a great motivator for me. But my history is I'm a basic scientist, so if I get too technical, please stop me and ask me questions. I will try not to do that.
Thank you.
And I will try to tell you a little bit about how I stumbled on RAN translation really before we knew what gene my husband's family had. And so let me get going here. So this is it used to be that if you think about the DNA and RNA and protein, and you get into science talk, one simple way to kind of think of it is that genomic DNA contains the letters of the genetic code, and it's double-stranded. And then there's RNA that is made from the DNA. And then there are proteins that can be made from the RNA. And it used to be that we thought that all of the proteins that were made were like sentences in this analogy. And each sentence had to begin with the word the, or there's a green light for a protein. And we all know that now that that's not true for repeat expansion mutations, and it's not true for C9orf72. It's certainly not true for sentences, that all sentences need to begin with the letter or the word the. And so what the discovery of this repeat associated non-AUG translation was that we discovered that the proteins could begin right with the word red dog, or it could shift a little bit, and there could be these unexpected sentences that shifted the letters that weren't understandable to us at all. So the proteins that we found are analogous to this, in that we used to think of these repeats that maybe they'd be in a protein coding region and they'd make a long sentence with a repetitive word. But then we found out that they can make these extra sentence fragments and mixed up sentences with scrambled messages. And those are like the repeat associated non-AUG proteins that we discovered. And so we discovered that in 2011. We were working on a really rare disease called spinocerebellar ataxia type 8. And so we reported this discovery, after working on this for quite a while, in 2010, early or late 2010, and then the C9orf72 gene was identified. And so I really thought that this is going to apply to C9orf72 ALS as well, and became highly motivated because we knew my mother-in-law died of it after the gene was isolated. So I think I'm going to be a mess. So there's a strong blow, and let's carry on. So it turned out to be correct that this was the case, that this DNA mutation, which is in a silent part of the genetic code, is actually producing two messages, one in the sense direction and one in the antisense direction, and produces these proteins, or these repetitive proteins. And so what we have been doing for quite a while now is to try to figure out how to make those proteins that are not needed and not expected go away, to see if we could develop some therapies. And we've developed two main strategies. One, we use an antibody approach to go after the RAN protein targets. And then the second approach is we use small molecules to turn down a pathway that we've discovered in cells is really important for producing these proteins. And so through these studies, which were both published in 2020, we've come to the conclusion that at least in C9orf72 ALS and our back transgenic mice, that it's these RAN proteins that are really driving the disease. So they're not normally made in people. So if we could get rid of them, that would be a great idea. So the C9orf72 expansion mutations in a part of the gene called an intron, it's caused by a G4C2 repeat expansion. It can cause ALS, it can cause frontotemporal dementia, and it behaves unusually in that it makes two RNAs, one from one strand of the DNA and the other from another strand of the DNA, and then makes these 6 unexpected proteins, which are called RAN proteins or dipeptide proteins. And there are many people that have been researching this disease like crazy, which is great. And some of the theories are that it's the C9orf72 gene itself and the protein that's normally made that's being reduced that causes a problem, or the RNA foci that cause a problem, or the RAN protein. So those are the main 3 things. And then there are all kinds of downstream effects when you start to look at the cell and things go wrong. You will find that there are many things that are wrong. So there can be inside different cells, you can not handle proteins well, you can have different kinds of stress, you can have mitochondrial function. And we're really interested in the upstream things more than the downstream things, because we think if we can control the really upstream toxic problem that the gene mutation is causing, we can prevent all the downstream things. And so the theory is like, if we could identify what we need to do to shore up the dam and prevent the flood downstream, we will prevent all the damage that occurs when a dam breaks downstream. So that's kind of our philosophy. And there have been a number of therapeutic strategies that have been used to go after these this particular mutation. One is to target the expansion RNAs. And there have been some really wonderful efforts made to get this to clinical trials really quickly. And Biogen and Ionis and Wave both performed clinical trials on C9orf72 patients. And the idea here that they had is that they blocked the expression of the transcript that is contains this G4C2 repeat. And the theory was that if they blocked that, they would also block the toxic proteins that are made from that transcript. And while that's true, a problem with that is that there's another transcript that's made in the opposite direction, and the antisense oligos did not block that other transcript. And so maybe for an antisense oligo approach and targeting the RNA and getting rid of the RNA, you really have to develop a method that will target both RNAs and not just one or the other. And what I worry about when I think about this, and other scientists worry about, is that, if the 2 RNAs may interact with each other, and we don't understand the normal biology of how this interaction occurs. And so if you knock down one transcript, you might actually inadvertently being upregulating the other one. And in some cells, that may lead to a toxicity. But in any case, the clinical trial that targeted one strand, those clinical trials were halted, and they failed. So we've been interested in targeting these dipeptide RAN proteins. And we know that from many studies that labs all over the world have done, that if you express these dipeptide proteins, they're toxic to cells and overexpression models. They are not expressed in healthy people, so they're really a great target. And are RAN proteins toxic in more physiologically relevant conditions? That's what we wondered, and we really thought we needed a model to test this. And so we developed a mouse model and published this mouse model in 2016. And these mice have the whole human gene, and they express both the sense and the antisense RNAs. So they're producing the 2 RNAs. They get these accumulations of RNA, just like the patients. They get the accumulation of these RAN protein aggregates, just like the patients. They have behavioral abnormalities that you can pick up that affect their gait and their movements, and they have motor neuron loss. And so using these 2 approaches, we had this multi-pronged approach to try to get at could these proteins themselves be toxic? If we could get rid of them, would that prevent all the downstream damage? And so in the first study, which I'm going to summarize next, this was done by Lien Wen in the lab and in collaboration with Neurimmune and with Biogen. And what she did was she took antibodies that Neurimmune had developed and peripherally injected them into our mouse model every week. And then at 43 weeks, she took down cohorts of mice that had the treatment or didn't have the treatment or were non-transgenic. And so here are the non-transgenic mice. They're mostly living at the end of her study. The C9 mice that have no treatment rapidly died. The C9 mice that received the antibody therapy lived longer. They weren't significantly different than the non-transgenic mice without the C9 gene, and did a much better, had a much better survival rate than the C9 animals that were untreated. When we looked at a number of different kinds of behavior, how the animals walked, there was always a difference between the C9 animals in black here compared to the non-transgenic or healthy animals. And then that was rescued when we gave them the antibody back or treated them with this antibody. And you can see on multiple types of this parameters from this treadmill test that there's a significant improvement with the antibody treatment, which is shown in pink. So a really important part of ALS, as you all know, is the loss of motor neurons in the brain and in the spinal cord. And in the spinal cord here, we can measure them. And in our non-transgenic mice, the level is here in green, and then there's a loss of these motor neurons in the C9orf72 transgenic animals, and there's a rescue of motor neuron survival in the antibody-treated mice. And a really surprising and exciting thing to us was that when we used the anti-GA antibody, not only did we reduce the GA proteins, but we reduced other proteins as well. So there are 6 different RAN proteins that are made with 5 different repeat motifs. And because the GA antibody or GA protein is a really sticky protein that traps other RAN proteins as well and prevents the garbage disposal of the cell from working right, you get this reduction of the not only of the GA protein that's targeted by the GA targeting antibody, but you also get this nice reduction of the GP protein and the GR protein. So there's this collateral benefit, if you will, to targeting and getting rid of the GA protein. So that was something key that we really learned, is that the GA protein is a really great target. So we were really hopeful that Biogen would take that into clinical trials because we thought that the data were really promising, but they did not take it into clinical trials. And so we went back to the lab, and we knew we had a great antibody to glycine alanine. And then Dr. Lisa Romano, who joined us with the big white glasses that are cool and have DNA on the sides, she figured out all the details of this antibody that we had, and then made a little transgene with it. And then we collaborated with another lab at the Children's Hospital of Philadelphia, Dr. Beverly Davidson's lab, to develop a gene that could be a one and done kind of treatment. So if the passive immunotherapy had gone forward, and maybe it still will, it would require a frequent drug administration about every 10 days. And for ALS patients, that's a difficult order. And with an AAV gene therapy approach, there's the potential for a single sustained dose that would allow a sustained expression of these antibodies in the brain. And that's the other thing, the antibodies, if you peripherally deliver them in the blood in patients, they really have a poor blood-brain barrier penetration, but the antibody levels are much higher if you can express the transgene in the brain and spinal cord. So that was the theory, that this approach could be a much better approach. And so I'm going to cut to the chase and summarize tons of work that Lisa and others in the lab have done, but mostly Lisa really led the charge on this. And this is a hanging wire test with a non-transgenic animal, and you see the non-transgenic animal going across the wire with no problem. The C9 animal has a big problem with this task, and we quantify all these behaviors. There's an antibody mimic that it's an antibody, but it's not to the GA proteins. It's an antibody very similar, and that did not make the mice any better. And then there's the antibody that Lisa worked really hard to express using this transgenic approach, and that animal does much, much better on the hanging wire test. And you can see that it makes it to the side of the. So again, to make a huge amount of work that Lisa's preparing now for publication, to summarize that, there were behavioral tests that she ran on the mice that improved. The motor neuron survival improved. The longevity of the mice improved. And this is a survival curve of the mice, the C9 mice that were treated with the control antibodies that are vehicle or control antibody, and the isotype control antibody, and they really didn't improve things. Oops. But then when she expressed the anti-G1GA antibody, the anti-GA antibody that she developed, the mice lived just as long as the non-transgenic and non-transgenic untreated or vehicle-treated animals. So this was really great, great news. And so a second strategy that we've been really pursuing is to turn off RAN protein production, because we think the RAN proteins behave differently than regular proteins. We need regular proteins in the cell to do the work of the cell, but we don't need these RAN proteins. They're bad proteins that accumulate. So is there a way that we could target those without targeting other pathways? And Tao Zhu worked on, who was the scientist that originally discovered RAN translation in the lab in 2010. He discovered that these RNAs can trigger a pathway inside the cell that leads to an upregulation of the production of these bad proteins and a downregulation of the normal proteins. And that pathway can be inhibited with a PKR K296R, which is a mutant version of this PKR protein, or with a type 2 diabetes drug, metformin. And that discovery was really great, because then that discovery was really exciting to us, because then we could test if metformin had an effect in cells and in mice, and it did. And it seemed to decrease the neuroinflammation that we saw in the mice, so it didn't have these angry glial cells. It improved behavior of the mice, so the non-transgenic and C9 animals behave differently, and then the metformin treatment improved the behavior, and in a couple of different, in a whole bunch of different tests. And importantly, again, there was motor neuron loss in the C9 animals that was rescued with the metformin. And so we have started, because we started a clinical trial, and are in the process of analyzing some of the data for the clinical trial now. So metformin is a first-line defense against type 2 diabetes. It's a drug that's been in the clinic around the world and in patients for decades. It's not completely safe, but it's pretty safe. So it's got a strong safety record, except for people with kidney problems. It penetrates the blood-brain barrier, and it's off patent, and it's really inexpensive. So so it has a number of advantages that would allowed us to then apply to the FDA to do an open label clinical trial. And because of the safety record of the metformin, we could go into the clinic pretty quickly. And the trial goals were to test the safety of metformin in this patient population, because it could be that patients with C9 wouldn't respond well to it, or would have some unanticipated side effect. And I can't tell you the results of the trial because we're still analyzing the data, but the safety profile of the trial is good. And so the second and secondary endpoints were ALS functional rating scale. The primary endpoints were safety, and then the reduction of these proteins in the blood and the CSF. And we're working like crazy to really get these assays to work so that we can measure these proteins. And we're setting up some collaborations to do that. And we're also really actively testing this. So the other trial goals are to see if we can get better at measuring speech, swallowing, and respiratory function, MRI, and other biomarkers. And to do this, we have assembled a team of fantastic colleagues. So Dr. James Weimer is the neurologist. He's performed all the neurological exams and the medical exams. Emily Plowman is a speech and swallowing and respiratory function specialist. Timothy Garrett is a metabolomics and drug and biomarker specialist. So he's looking at metabolomic changes in the blood and the CSF. And then David Valancourt has been doing MRIs to see if there are cortical spinal tract changes in these ALS patients, or if anything slowed down and changed with the treatment. And so all of these measures are probably beyond the goal or what we are going to be able to measure in this open label clinical trial. But we think that through this experience, we're going to get better at doing a clinical trial. And if successful and we get some hints that this is working, then we're going to go forward with a placebo-controlled, much larger multi-center clinical trial. So the experimental design was to escalate the dose of metformin over a 4-week period, and then do the study out to 24 weeks. And we drew blood and CSF at each of the visits, so before and during the course of the treatment, and then there was a follow-up study out to 52 weeks by telephone. So we've completed the recruitment, and we're now analyzing the data. And as I said, if promising, we will go for a larger future placebo-controlled clinical trial. And we're excited about this because we know that these RAN proteins are not only in C9 ALS, but we discovered them first in spinocerebellar ataxia type 8. And they've been found in Huntington's disease. They've been found in a number of the other ataxias that we're working on publishing now. So they've been reported in 12 different diseases in publications, and there are about a half a dozen more that we know of in the lab, in our lab and other labs. And so we wondered if RAN proteins could be found in sporadic ALS cases, because if they could, then maybe we could target the RAN proteins and we could get rid of them. And familial ALS, we know about 60% of the familial ALS is now explained by known genes, but only about 10% of sporadic ALS is explained by known genes. And if you think about sporadic ALS, 90% of the patients that come into the clinic have sporadic ALS with no known genetic cause. And if you have no known genetic cause, it's really hard to participate in a clinical trial because you don't know what your enemy is, you don't know what to target. And so we wondered if RAN proteins are found in sporadic ALS cases, and we looked at sporadic ALS autopsy brains, and we found that 10 of those brains, or 10 of 45 autopsy cases, or 22% in this initial study, who were negative for the C9 mutation and also for SCA36, another expansion mutation that can produce RAN proteins, were positive for 2 of the RAN proteins, anti-PR or anti-GR, so proline arginine or glycine arginine. So these are proteins that are also found in C9, and we had the antibodies and we're good at looking for these. And so when we looked in sporadic patients, we were really surprised to see that so many of them, those autopsy brains, also had an accumulation of these proteins. So then the next question became, could we use this information to identify the gene mutation, and would that then allow for new therapeutic opportunities? And so Dr. Leanne Wen, who was in the lab at the time, but now has her own faculty position at the University of Florida, developed a way to pull out, so use this information about the aggregates that were found in the autopsy brains to design guide RNAs, and then to use a broken form of CRISPR-Cas9, which maybe you've heard of, can cut out very specific sites of DNA. And in this case, we used a deactivated version that would not allow cutting. So the sequence information from the aggregates was used to predict what RNAs might be, would allow production of the GR-containing proteins. And the idea is that if somebody had this kind of GR protein pathology, that then we would hypothesize that just like C9orf72, they'd have a long repeat in their genome somewhere. And if we could use the deactivated CRISPR system to pull it out, we could enrich for things right from patient DNA that would give us a clue to what was causing these proteins to accumulate. And we put a little handle, a biotin handle, and a magnetic bead to pull out the repetitive DNA with this method. And in contrast, we don't pull out the normal length repeats from control individuals because they're not as long. There aren't as many of these complexes that can sit down on the DNA. So it's like an enrichment trick or a needle in the haystack to pull the gene of interest right out of a haystack with a magnet. And Leanne used many different techniques to really hone in on look at this DNA. And once she had identified the DNA, then she could go right to figuring out what gene was a problem. And this is not a sporadic ALS case, but this is a autopsy image from a patient with sporadic Alzheimer's disease. And she designed these guide RNAs and used this dCas9 read approach to pull out this long repeat in the gene from the genomes of these Alzheimer's patients. And she found a GG-GAGA expansion, and that GG-GAGA expansion, a specific variant of it, increases Alzheimer's risk. And she just published this. We just published this last week in PNAS. So we're really excited about this paper, and we're really excited about this because we can apply the same thing to the patients that we've found these aggregates in that have sporadic ALS that we don't know what genetic cause of ALS they might have. And so what we're really hoping to do now is use this idea to do translational medicine where we can, patients always tell us what's going on. Patients are the experts. You all know that. And when physicians see patients, they can draw blood samples. Sometimes patients will generously donate their brains for research after they pass. With the blood, we can make iPSCs, we can make organoids, we can look at autopsy tissue, we can detect these aggregates in brains, we can use that information to identify genes, and then that increases therapeutic options. So including maybe antibody treatments and also treatments potentially with metformin. So I want to shout out to Lisa Romano, who's a fantastic scientist in the lab that it's just been a great pleasure to work with. Tao Zhu, who's worked with me for a long time, initially discovered RAN translation. Leanne Wen, who's gone on to start her own lab at the University of Florida, and then Xu Guo and Lisa have also been working really hard to better understand the genetics of sporadic ALS. And then our colleagues and funding agencies, members of the Center for Neurogenetics, where I work, and really a huge thank you to all the ALS patients that participate in any research. And I'm really happy to take any questions that you might have. So thank you for your attention.
Thank you so much for such great, presentation. And obviously it shows that how much work you've done, and also how you brought yourself both to your emotionally and mentally in here for ALS. So can't thank enough for all your dedication and your energy, because this is how we're going to solve it. It takes all of us, and
yeah, we're all motivated, aren't we?
Very motivated. So with that, I am joined here with my colleague, Natalia, and we will moderate the questions. So we've had a lot of questions, so we will continue to ask. So Natalia, I'll kick it off with you starting the Q&A.
Yes, I think we had the first question in the beginning of the talk, and you kind of went into more details about the differences between RNA and RAN. But for some people who joined later, if you can talk a bit more about it, and as well as toxicity of both, and how you were kind of starting your presentation as well.
Yeah, so DNA is double-stranded. DNA makes two RNAs, and then we used to think that proteins could only be made if they had a certain start signal, and or an AUG initiation codon. And the analogy that I used is that's like thinking all sentences. We have these letters of the code that are the DNA level change a little bit in the RNA, and then they can make words, and all of the words in the protein language are 3-letter words. It's not 4-letter words, it's 3-letter words. But and we used to think these proteins all had to begin with a 3-letter signal, AUG, or the word the. And now we know from work that we've done in the past that that's not at all true. For these repeat expansion mutations, they mix up the protein producing machinery. And when they mix that up, these unanticipated and unwanted proteins are made, and they muck up the brain. They accumulate in cells, and they cause a toxicity in the brain, and we can get rid of them with pharmacologically with a drug. Metformin reduces them. We're trying it. I'm not recommending that everybody go out and get on metformin. I think we have to do a really, we have to do a placebo-controlled trial to know if it's going to work or not. And we have a ways to go, but we're doing that. And we're really excited about metformin because it is an FDA-approved drug that could be, can be more quickly studied because it doesn't have all these toxicity studies that you have to do. We just have to test if it's safe in patients and if it's effective in patients, and then do a placebo-controlled trial. So we've got some things in the pipeline with antibody therapies that make our mice better. Metformin makes our mice better, and we've got a clinical trial that we're doing on metformin.
Thank you, Laura, for
Yeah.
that detailed answer. One of the questions that people had was, is it possible to crush metformin for use through feeding tube?
We didn't do that. I'm sure you can crush it and put it in a feeding tube. I'm not sure if a doctor would recommend that or not. What we used in our clinical trial, we thought that would have to be tested. Different formulations would have to be tested. But what we used in our clinical trial is a version of metformin that's a slow release metformin. And so we liked the idea of the slow release metformin so that it would have maybe a prolonged effect over time. But it's a good idea because in our clinical trial, one of the requirements was that a person had to be able to swallow pills. That excludes some people. And I think more research is needed to answer that question.
Thank you.
And I'm a Ph.D. doctor, so you can ask a real doctor.
Next question, we have a bit more detail about metformin. If there is like any data, or you can look back and see if there were any presymptomatic C9 involved in the clinical trial of metformin, or if there is any way to kind of assess for, as it is the first line of treatment, if there are any presymptomatic C9s who were taking metformin based on the past records.
There have been a couple of interesting studies that are really large epidemiological studies that have been done in the past, not really asking that specific question. But there have been a couple of really big studies looking at type 2 diabetes and concluding that that was protective against ALS. So they didn't get down to were they using metformin, but metformin is the most commonly prescribed drug. So it's very possible that could have been the explanation. And then initially, when we only knew that there were RAN proteins in C9 ALS, and that seemed to not really explain a lot of ALS. I mean, it explained a lot, but not maybe enough patients to get a signal like that in an epidemiological study like that. But now that we know that there are some patients with sporadic ALS that have other, they don't have C9orf72, but they have RAN proteins. I mean, it's possible, but it's hard to tease that apart in these big clinical data sets. It's hard. The data are noisy. It's hard to go back and figure out who's pre-symptomatic, what stage of disease they're at. It's just, there's not enough information. So I think we have to do a prospective study and a clinical trial to really nail it one way or another. And I think it's really important to nail it so that we know if it does or doesn't move the needle on ALS and C9 patients, or maybe in the future, carriers or these sporadic patients that are RAN positive. Because if we know it does move the needle, then we can really recommend it and a lot of people will get on it. If we just have it be kind of a rumor, people will, some people will try it and lose interest. So Lyle Ostro, I see he's got a comment about metformin, maybe.
No, I'm not a real doctor, but you asked for somebody to comment. So the release version can't be crushed. Real doctors are PhDs, well, anyway, but it can't be crushed, the extended release version. But I'm sure somebody will look at that.
Yeah.
Thank you, Lyle. As always, very helpful. Is metformin useful for people with ALS with only C9 or other types of ALS as well?
Well, we don't know yet. We don't know yet if it's useful for people with C9. We're testing that. It's useful for our mice. We have to prove it in the clinical trial.
Yeah.
Our hope is that it might be applicable to patients with C9 and also with other RAN protein diseases that have yet to be discovered that cause ALS. So we have this RAN protein signature in quite a few ALS patients that we've looked at. And so it might similarly decrease RAN proteins in those other diseases. When we look at other repeat expansion mutations that cause myotonic dystrophy type 2 or SCA8, metformin does work. It makes our SCA8 mice better too. And so we think, and it has made Huntington's disease mice better. They haven't looked at it from a RAN protein perspective, but I think that there's great potential for this to go across the RAN protein diseases, and that there are other sporadic ALS RAN protein diseases out there to work on.
Thank you, Laura. And one question that I missed earlier was, what was the dosage that was used? I bet you talked
started patients at 500 milligrams once per day, and then for a week, and then went up to 1,000 for a week, and then went to 1,500 for a week, and then the fourth week we went up to 1,000 in the morning and 1,000 in the evening. And most patients tolerated it well. Some patients have an upset stomach, so that's a common side effect. There can be serious ketoacidosis. So if you do take it, you need to be monitored, and you need to test. You need to talk to an MD doctor before going on it, and so they do the right tests for monitoring that.
Thank you.
But I'm really excited about figuring out if it really works or not in people. I think that's a missing piece right now, and a piece we're very excited about figuring out.
And a lot of the aging research, right? People are doing a lot of aging on metformin, and
well, and with our
people are getting on it. I'm on it.
With our new Alzheimer's study, we found a repeat expansion mutation that produces these RAN proteins. And we don't know, we don't have a mouse model of that yet. Dr. Wen is working on that. But what we do know is if we treat cells with metformin, it helps the cells. And so it's also reducing the levels of those proteins. And they're different proteins. They're hybrid, dipeptide hybrid proteins.
Yeah. Thank you.
Thank you. Next question we had about long-term immunogenicity of AAV-mediated delivery, which you're using, and if you have any thoughts on that.
Yeah. I'm kind of new to the AAV field, so my colleague Bev Davidson could probably answer this much better, but what I hear her answer to that when I've asked her is that the newer capsids are much more effective at targeting the right part of the body and the right cells in the body. And so that's why we're collaborating with her to really try to get a capsid that will hone in on and get these antibodies across the blood-brain barrier into the right part of the body. And then it's important to have the dose as low as possible. And then, there are some potentials for changing the capsid, which might decrease immunogenicity, and the viral capsid that delivers the gene therapy. And also there are some drugs that can suppress immunotherapy. If you've had previous exposure to AAV, that's more of an issue. But I think it's an issue that the gene therapy community is well aware of and has some tools in the toolbox to make it worthwhile.
So the next question is, how can one participate in the trial?
In
the metformin
trial. Well, right now we're not recruiting patients. we hope to be, if we have positive results, we hope to launch a bigger multi-center clinical trial, and we will post that on clinicaltrials.gov, and we will be working with our colleagues across the country, probably including Lyle, and so then you can people could participate in it then that way.
And it will be for people with C9 repeat expansion.
The first trial would be for the trial that we've just finished, the open label trial that we're trying to finish analyzing the data for is in C9 patients. And so that would lay the groundwork for a bigger C9orf72 clinical trial, would be my expectation. But I think that that could expand to pre-symptomatic carriers if we have good biomarkers in the future, sometime in the future. I'm not sure when in the future, that's not next up. And then maybe there would be some applicability for other patients with RAND protein diseases that cause ALS that are not, or whose genes we haven't discovered yet, but we're working on it.
So we are working on asymptomatic research where we actually do, a digital biomarker monitoring for asymptomatic patients. So
yeah, I think that's wonderful work, really important work, and I think that early treatment is going to be key.
Yeah.
Can I say something that I think I really want to? Yeah. I think it's really important. So, first of all, this trial that Dr. Ranum is doing, the metformin trial, was funded by the Department of Defense, the CDMRP ALS research program. And the fact of the matter is that funding is thanks to you guys, right? Like, yeah, the whole ability of that Department of Defense program to fund clinical trials are expensive, was thanks to the advocacy and the loud voices of people living with the disease, ALS warriors who, advocated in Congress to get that funding approved. And so the trial would not even be possible if it wasn't for you guys. And when somebody was just asking about, like toxicity or immunogenicity of these things, so when there's a cool new idea from Dr. Ranum's lab or different labs in ALS research, there's a lot of things that have to happen before it can be in a clinical trial. Now, metformin is super exciting, right? Because we already know it's safe and we use it for other things. And that's a unique situation that makes it exciting. But part of the process, there's something called IND enabling studies. So if there's a new treatment, like an AAV vector or a vaccine, these different sorts of treatments, there's a whole lot of work that has to be done to make sure that it's safe, right? And in the case of these AAV vectors, sometimes we worry that the AAV itself might cause a problem, or the payload that it's delivering might cause a problem. But all of this is studied in different animal models and in very, very detailed way. And the reason I want to bring that up is because we need your help again, guys, because, I'm sure everybody knows what's going on in the world right now. Those studies, this is the purview of these IND enabling studies, are what FDA requires to make sure something is safe and to approve it for a clinical trial. And science matters. And right now we're in jeopardy of kind of losing the ability to move this sort of thing forward, both from funding these IND enabling studies and the early science, but also regulating at the level of the FDA to make sure this stuff is safe. And so, it's your voices that, again, are going to make a difference, just like it did to get this kind of thing funded in the first place.
Yeah, absolutely. And metformin it's cheap, and that's great for patients. It also makes it, ironically, harder to do a clinical trial because no pharmaceutical company wants to pick it up. So we depended on funding from both the ALS Association, which we're tremendously grateful for, and the DoD funding. And it was those two funding sources that allowed us to do this. And so it's donations to the ALS Association, but also it's your taxpayer dollars and advocacy, patient advocacy. And so that I echo Lyle's comment that this is just so important, right, especially right now. We don't know what changes there are going to be, but patients don't want to have drugs that they test on themselves without a really rigorous protocols in place to do it as safely as possible. So we need the FDA, we need basic science, and we need your help because you have the most important voice.
Thank you.
Thank you. Next question we have is if iPSC cells derived from C9 patient produce the same profile of RAN proteins?
Yes, they do. So Lisa has done some of that work, and they do. And so iPSCs and iPSCs can be differentiated into neurons or organoids as well, and we can see the proteins there.
Cool. So another question was, is there any research from the RAN protein perspective that you have noticed a trigger that causes an individual from asymptomatic to symptomatic?
Well, the pathway that I was talking about, the PCARE pathway, is a stress pathway. And it's an important pathway in cells that if you have a fever, you can trigger this pathway. If you have a viral infection, you can trigger this pathway. If you have the accumulation of proteins, you can trigger the pathway. So you can trigger this pathway in a number of different ways. And so kind of the overall concept is that I think there is very likely a stress that triggers ALS and triggers C9 ALS. Patients are healthy for, they might get it at 10 years of age, but they might not get it at all. They might not get it in their 90 and die of something else. And because C9 is a leading cause of sporadic ALS, that means that, this gene is in the families and doing something. And so there must be some triggers. And so I think some common themes of things that I think of from my neuroscience background is inflammation is bad, so you don't want to have inflammation. You don't want to be stressed out. You want to do things to live the best life you can live and not trigger the production of more of these proteins. So sometimes I think injury might be a trigger. So there are investigations ongoing into that. Does injury trigger the production of these proteins? And so I think that there's an environmental component in this that's maybe really makes the age of onset and what you're going to get, where the onset's going to be, depend on what's happened in your life. How you've fallen. Have you fallen on your head? Have you fallen somewhere else? Have you done something
from peanut gallery about that again, because fact is the only way we're going to figure that out is if we can figure out when people are becoming symptomatic, right?
Yeah.
And that's why what Everything ALS is doing is so freaking important, because right now, we don't have good ways to detect these changes early in disease. Then you have to lose a certain amount of your motor neurons before you even notice that you're weak. And it's these, trials that Everything ALS is doing and some other organizations looking at, can we really tell in a very sensitive way early on when somebody is starting to what we call phenotypic conversion, which means going from not having symptoms to having symptoms. And that's critical, because until we have those tools, we're not going to be able to test things to see if we can affect those early changes.
Right.
Thank you. We have next question, quite personal, but if one has
stress in this moment,
has sporadic C9 ALS and already taken metformin, would your recommendation be to continue doing it or wait for the clinical trial results?
Okay, well, you know my situation. My husband has ALS, and so we were faced with this. We had not finished the mouse study, and then we just had to decide if it was, potentially worth a try. And so I can't make that decision for anybody else, but I can say that I think, because it is an FDA approved drug, there is the potential that you could do this off label. I don't know if it's a good idea or a bad idea at this point, because we have to do the clinical trial. But I think that's a personal decision that everybody has to weigh and face themselves.
Thank you. Although this is, we're talking about the ALS study, are there any markers in evaluating FTD symptoms, either in ALS or early FTD symptoms?
Yeah, ALS, in C9, there's a clinical spectrum. So some of the assays that we do in our mice could be FTD relevant, open field behavior and different things. We could do more in that realm than we have, but yes, people are definitely looking at FTD and ALS. Just because the disease ALS alone is so complicated and it's so hard to measure, the clinical outcomes, the ALS FRS rating scale tries to sample all the different things that can go wrong in ALS. And so we focused on C9 ALS patients for our clinical trial, just to make it easier to. But I think, both are very important, obviously.
Thank you. Next question is about NFL levels and how they might determine the phenotypic conversion in C9.
Yeah, there is a wonderful study out of the University of Miami that's looking at pre-symptomatic carriers where there is a signal. Michael Benatar's study showing that NFL does increase as people phenoconvert. I've seen him present those data at scientific meetings, and maybe Lyle has more to comment on that. But I think it's a great marker. In the clinical trials that failed, the NFL levels were going up, and it was a signature that things were getting worse. In the spinal muscular atrophy clinical trial that really helped SMA patients and those SMA kids, the NFL levels went down. So I think it really is a good marker of neurodegeneration, and something that we'll also be looking at in our clinical trial.
The next question is there a way to reduce RAN protein naturally without metformin? By diet, by controlling through diet?
Well, I do think, I don't know, because those studies are really hard to do, but when we knew Bert had C9orf72 ALS, and the gene really early on, we did everything we could to change our diet to be a low inflammation diet. And I think maybe that has helped. He's now almost 9 years into the disease, and nobody in his family went more than about a year and a half. So he had 8 other family members die. So I don't know what it is about him. He might just be extra tough because he had to live with me so long. I don't know. or it could be some combination of things that he's doing. But I think, I'm really interested to learn more about all the tips that your community has, and to read McFinn's book on what he is doing. So.
Yeah, that's amazing. You described some ways, or like in the lab, to kind of measure things. The question, I guess, is when or how is it possible to measure like RUN protein from patients?
Well, there are assays that we're working on developing where we can take a biosample and then capture the protein with one antibody, and then once that's captured, sandwich it with another antibody that emits a signal. And so with these antibody assays, they can be really sensitive. They're hard to develop, but once you have them, they're really powerful ways that you can measure the levels of the proteins. And so Lisa Romano has been working hard on developing that with Tala Rippin in the lab, and we're looking forward to collaborating with Len Petrocelli and his group too, to measure some of these in the CSF, because he has worked that out for some of the CSF, to detect some of the RAN proteins in the CSF samples. And we think measuring them in the CSF will be really important, because that's, behind the blood-brain barrier, may be more reflective of what's going on in the brain. But we don't really know, getting back to the NFL and that being a biomarker that increases when you have phenoconversion, it could also be that these RAN proteins are going up when you have phenoconversion. So there's a lot more that needs to be developed, and assays to measure these proteins that will be really interesting to see what the natural history is of these proteins. It could also be that, if you start to get rid of some of the aggregates, that the measurable RAN protein levels go up. So we need to do these studies also in mice, where we can, harvest the brains of the mice and correlate that with what's going on in the CSF.
Thank you. So the next question leads into what you were saying. What do you think could be the mechanism of action of metformin in C9 patients? The RAN protein, anti-hyperglycemic effect or mechanism, AMPK effect, and mitochondrial effect?
Well, it could have effects on all of those things, I think. But it does have a direct effect on the protein kinase R pathway, which is this pathway that's really important for regulating RAN translation. And so we're doing studies to try to understand all of the effects and why the RAN protein levels go down. But we also know that we can just very specifically inhibit the PKR pathway with the dominant negative PKR, and we get the same decrease in RAN protein levels. And when we treat with metformin, we also get the same reduction of the phospho-PKR. So we are inhibiting this pathway that we know is really important for producing the RAN proteins with metformin. But it could be that it's a combination of other things too.
Can I ask a question?
Yeah.
Yes.
So, Dr. Ranum, can you comment about sort of the selective vulnerability? I mean, if somebody has a C9 expansion, that's in every cell in their body. And so what is it about the motor nerves or even nerves? Like, I mean, are there RAN protein aggregates in my, skin and in my pancreas? And if so, what's the difference?
It's really a great question and a real mind teaser. We don't really know. So in our mice, when we looked at where the RNAs are accumulating and where the RAN proteins are accumulating, we found something kind of striking, which is that the sense RNA is everywhere, and the antisense RNA was in the motor cortex and the spinal cord. And so it could be that when the ASO treatment is targeting just the sense RNA, that the antisense RNA goes up and causes a problem. And that might be, they might regulate each other just in the places where they're both expressed. And if you have an antisense RNA, you're going to get different flavors of RAN proteins, including the PR and PA proteins. The PR protein is at least quite toxic to cells and model systems when you overexpress PR. But I don't know if I think just a lot more needs to be done. You know, these weren't found that long ago. And so we have to build all the tools. Every time you look at a different tissue, you have to optimize it and figure out how to really assay the proteins there. So where they are, we got a lot of work to do. We and others, the big we.
Thank you. Another question, I think, talks about progranulin for C9 FTD patients, that there are some clinical trials on that, and whether you think it might be helpful for ALS at some point as well.
Maybe Dr. Ostrov can answer that. I'm not sure. Progranulin treatment for C9 patients?
Yeah, I think Thomas was asking this question. Yeah, I'm not sure.
Yeah, not quite sure what's being referred to. So that's a different mutation that also can cause FTD. I'm not aware
Yeah,
of.
Maybe it's just a different treatment for a different type of ALS.
Or FTD too. So it's
or
a
FTD.
mutation that's found in people with FTD. And and somebody asked about. I saw a comment that was really insightful in the chat there about if the treatment works for ALS, it's probably going to work for FTD. FTD is even harder to measure, and it's even harder to know when it starts. And there's I'm sure people on this call know, like, a lot of people who get FTD initially are misdiagnosed, and sometimes it's another kind of memory trouble. Oftentimes it's actually marital problems. So people with FTD sometimes first present to a marriage counselor because of the
wow.
behavioral changes, and so it's really even harder. The change has been that now that we're testing people for these genes that cause both of these diseases, and by the way, we're able to do that because the gene tests are sponsored by the drug companies, because they want to make sure that they find people who might benefit from their therapies. So now we have people who we know are at risk, have the expansions, and we can follow them more closely, and hopefully we'll figure that out. But FTD is even harder to measure than ALS.
So I see a question in the chat here about the anti-inflammatory diet. So there.
Yeah.
That
was going to be my last question to you. Can you share, you mentioned about your husband being on anti-inflammatory diet strategy. This would be a great way to end this talk by getting something actionable for people that you can share.
Yeah. So what we did, what I found really useful is our books by Mickey Trescott and Sarah Ballantyne, and I can type them in the chat. And Sarah Ballantyne has a Ph.D. and she had not ALS, but she had some inflammatory disease, and she just had a real hard time with her health. And I think a lot of people have a real hard time with their health. And she kind of broke it down to all the things that you eat and what their biochemical effects are on cells. and which foods can affect the gut and the barrier that your gut has between your gut and your body. Because if you have a leaky gut, you have a lot of inflammation. And so she has cookbooks, and Mickey Trescott as well has cookbooks, and there's a lot of information out there that you can find these books, and that's what we did. So we kind of stopped and changed our diet on a dime. So we just stopped eating bread and things that were on Sarah Ballantyne's bad list, with the theory that maybe it would be helpful. And I have some sort of gut problems, and so I felt like I was the canary in the coal mine. If my gut was better, then that was probably a better diet for my husband. And but anyway, I think that probably did have an effect too. But all these things that you need, maybe 100 years to prove, when you have ALS, you don't have that kind of time. You have to decide what you're going to do. And so that's one of the things that we decided to do, change our diet.
And I see that the Paleo Approach, Reverse Autoimmune Disease and Heal Your Body by Sarah Ballantyne on Amazon, and the Nutrition Dense 125 Autoimmune Paleo Recipes for Deep Healing and Vibrant Health by Mickey Trescott. Okay, perfect. Thank you very much. What a great way to, share your insights and your personal story. And thank you for bringing yourself to this event today. Really means a lot. And, we wish you good luck as well as you're going through, and hopefully we can help you and your journey. Yeah,
well, I'm excited about that. And I'm going to pick up the book, and my husband's also written a book. It's called Clinical Trial by Robert Ranum, if any of you are interested in that. It kind of talks about some of these things that we did early on, and he's still here, so he's still walking and talking, and you might find that interesting too.
Yeah, and we would love to chat with him and you sometime, so
yeah, that would be great. Okay. Well, thank you very much for including me in this. It was a great session.
Thank you for giving us your time.
All right. Take care. Bye.
Have a great day. Bye-bye. Thank you so much, Lyle. Okay.
All right.
It's all yours.
Thank you, everybody. The Pathfinders are here for you. There are 11 of us. You can find us at the App Store and the Google Store. Take advantage of it, because our hearts are in it, and our minds are working for each of you.
Thank you.
So, folks, what a pleasure. What a pleasure. Lyle. Good night, Javier. All right. I'm going to press the button, folks. We'll see you all soon.